Ladies and gentlemen, this is a privilege to have a chance to show some of the project results to this audience. And I selected a specific approach which I never used before, because I never had a chance to flashback 20 years when the project started, and after 20 years to see what is the consequence, what is the outcome, and what was actually the impact of the research we did in the frame of this project. As already mentioned, this is the list of project members, the main groups cooperating in this Copernicus project. I was a little bit surprised that some people don't know what is Copernicus, because in this society of equine researchers, Copernicus project is really a term which almost everybody knows, because numerous publications were made in the frame of this project. So the main driving force was coordinator of the project and his group in Vienna, completed by Thomas Rummel, Johann Söldner, and Robert Achmann. Then the second group was a group in Hungary around Professor Imre Bodo. The third group was our group in Ljubljana. And then the Swiss group, Professor Lazari and Elian Marti, and certainly support of LIF as an organization for all our field trips. Not at least, it was important that in the project were involved 12 PhD students. And these PhD students did a huge part of the research. What were the objectives of the project? First, to comprehensively document the lipids and breed. Then to collect phenotypic data from almost all living horses in the stats we visited. To reveal genetic structure of the lipids and population. To estimate immunogenetic parameters, because we were kind of a transition period from classical biochemical immunogenetic markers to molecular markers. This project was actually one of the first projects where molecular markers were heavily explored and used for the description of a breed. Then to compare pedigree data and DNA data, to examine general health status of the breed, and to produce documentation material about the breed. And we really collected a huge photographic and video library, which is still available and can be used for further research. And not at least, we wanted to promote lipids and breed through scientific publications, because we believe that good reputation in scientific journals increases the impact and value of the breed, although we never doubt on the value of the lipids and breed. In this slide, I have to apologize for politically not up-to-date map. You see there were several political changes since this map was drawn. Anyway, material was collected during 2 years, 97 and 98, and at this time all these 11 stats were visited, animal measured, blood collected, photo documentation archived, and this was the base of the material which we then used for our research. What were deliverables of the project? First, extensive set of data for morphological traits, large photo and video documentation of all animals, and not at least, methodology for analysis of phenotypic traits using photo documentation. Hungarian group was very active at that time in this part of the project, and now Thomas, I see, is taking over and improving this research. Now I will walk through several articles which were published directly from the project. And certainly the first paper was morphological description of the Lipizan horse population. And in this frame, a methodology of taking 37 phenotypic traits from a single horse was established and used through the whole project. During the project, we were drawing many trees, phylogenetic, some clustering trees, and you will see more such trees during my talk today, and they are based on different data sets, and this tree represents similarity of horses in different stats based on morphological traits. This is a dispersal of phenotypic traits which we got after principal component analysis, and you see that animals from say classical stats, Lipica, Piber, and Topolchanki clustered quite close together on the upper left part of the diagram. On the upper right side are animals from Silberschwarat, because the stat followed a little bit different selection strategy. And in the lower part of the graph are animals from mainly Romanian stats. This is based on the data for mares, similar for the data of stallions. You see some differences in morphological traits between stats. The next type of analysis was analysis of diversity and population structure in the Lipizan horse breed based on pedigree information. This was quite adventurous part of research because documentation was not available on one side, so we have to collect these pieces of information in different stats. Part of the documentation was in Italy, in Monterotondo, and then were made copies of these stat books, and pedigrees were reconstructed. And these are the first results based on this research. And we were able to network all these animals into the single pedigree. From the pedigree directly, we can estimate the level of completeness of this data, and you see the thick line is the average completeness of the data, which is quite good until the 10th or maybe 15th generation, and then drops down with further generations. A little bit lower quality of data was found in the Romanian stats, and maybe a little bit better in Monterotondo for a certain period, but on average, pedigrees were complete somewhere between 10 to 15 generations back. Also from the pedigree, we can read what was the impact of certain breeding animals and certain breeds on the establishment of the Lipizan population. And you see that Baroque horses, either from Spanish or Italian origin, take more than half of the gene pool. Then the next important piece are Arabians. The old Lipizan genotypes represent about 10%, and then also contribution from Friedrichsburg and Clatterby and English and Shagia Arabians. A little surprising story is that among founder stallions, there are only 2 with large contribution, which are still present as founders of today present stallion lines, and these are Toscanello Hedera and Neapolitano. All other contributors don't represent classical stallions. What is the estimated frequency of the most influential animals in the pedigree? You see that the majority of these animals was born somewhere in the 19th century. And on the lower part you see the most important founders for the population in Piber and Giacomo, and again a tree which clusters stats according to the contribution of different founder animals in today's population. This is a part of immunogenetic research where genotypes were established for the ELA locus, and you see that there are some differences between stats, and based on these results we looked for heritability, for IgE concentration against different environmental allergens, and there were two which were studied in this frame, Alternaria and Aspergillus. And you see that heritabilities for these two antigens are respectable, 0.33, 0.34, although for the recombinant antigens the heritabilities were much lower. Now regarding the environment where the animals live, you see that the influence of the stat is significant in the case of rheumatoid arthritis for both antigens, Alternaria and Aspergillus. Then we move to mitochondrial DNA. We try to reconstruct the history of maternal lines using the empty DNA data. We sequenced mitochondrial D-loop, and we found in the population of Lipizans many more maternal lines than only traditional maternal lines which were established in the state of Lipica. And we ran out of clever names, so we just labeled mitochondrial haplotypes by letters. So these letters on the right side of the table represent newer maternal lines which were mainly established in the eastern states like in Romanian and some Hungarian states. And the mitochondrial types having nice old-fashioned names are mitochondrial types which are represented in classical maternal lines. The variability on the 5' end of the mitochondrial DNA was much greater than on the 3' end, and then we started to look for scenarios how during the evolution of the breed these haplotypes may develop, and this is shown with these networks here in the center of the graphs. And if we cluster mitochondrial haplotypes in the Lipizan breed, we end up with the same distribution as in majority modern breeds. We have kind of 4 groups which are present in almost all modern horse breeds and also in the Lipizan breed. Then we moved to microsatellite analysis. We estimated the heterozygosity in the breed, we estimated the allelic richness of the markers, and both parameters were quite comparable with other modern horse breeds, so no trace of outrages in breeding in Lipizan breed, although the population is relatively small, and it could be assumed that inbreeding is higher than in the bigger breeds. This is a consequence of the classical, wisely selected mating scheme which conserved stallion lines and maternal families. However, in Lipizans we found 2 loci, microsatellites, where heterozygosity, which we observed, was much lower than expected. And what was the reason? The reason was that even in this set of microsatellites, which are routinely used for paternity testing, there were mutations where the primers align with the genomic DNA, and this is the reason for null alleles in Lipizans. So in these 2 loci, which are labeled with the red arrow, we have a higher proportion of homozygotes in Lipizans than in other breeds, but this is This is for the technical reason, not for biological reason. These numbers illustrate the microsatellite diversity across different populations, and you see that average heterozygosity is between 0.65, 0.70, which is a reasonable figure for horse populations. We analyzed with a little bit different software this data and show that there is a considerable overlap between Lipizan populations from different studs. However, in this case, Kladruber horse is distinctly different population, and this can be shown even more impressive maybe on this graph, where you see this compact cluster in the middle of gray animals. These are Kladruber horses, and also on the right side of the graph you see separated Kladruber population from the other Lipizans from different studs. Each color is a different stud which are grouped together in a cluster. And from this work we actually made some follow-up study which was published right at the beginning of, at the end of this year in Swedish Agricultural Journal together with colleagues from Slovakia. So this research still has some impetus which is leading to the recent publications right now. About inbreeding microsatellite heterozygosity, morphological traits in Lipizan horses reported this article. The same set of data, 18 microsatellite loci, were genotyped for all Lipizan horses. Inbreeding coefficient was estimated from genetic data. We had first estimation from the pedigree data, which was around 10%, and inbreeding coefficient from microsatellite data showed the same value. You see that even during the end or at the end of the project, some our articles made it to the cover page. And this is a special honor for authors when their articles convince the editors to put the object of research on the cover page of the journal. And this is one of such examples. Journal of Heredity published it. And as already mentioned, all this work was gathered together and summarized in this nice book, which is probably the final result and final product of our project. Then I added some follow-ups. We used Lipizan story to show how we can use these new genetic tools to manage populations, and this is such an example from our group. Then there are several articles using mitochondrial DNA as a molecular marker for population analysis in other Lipizan populations, in other breed populations. So there was a number of articles with this topic. Then we have quite a recent study, comparison between manual and stereo vision body traits measurements of Lipizan horses, bringing new technology, new approach into the measurement of phenotype. And just a recent publication from Thomas, Max Doblichberger and Gottfried Brem, rating the equine conformation, just published this year. As already mentioned, this is probably one of the highlights of our follow-up studies of the project, a native genetics study about graying. And Leif Anderson added his knowledge and his passion actually for simple monogenic traits in farm animals. For all those you maybe don't know, his group is kind of specialized on simple monogenic traits which are of relevance for farm animals. And they looked for the speed of graying, for incidence of melanoma, appearance of vitiligo, and speckling pattern in horses. And the next follow-up was article on complex inheritance of melanoma and pigmentation of coat and skin in gray horses, which also had kind of relevance for medical studies, for studies of melanoma in humans. And you see here, estimated heritability for melanoma is about 37. It's really high. For graying, certainly because it is a monogenic trait, is almost 0.8, vitiligo 0.64, and speckling. These are all simple monogenic traits. However, melanoma is not monogenic. Melanoma is polygenic trait, and therefore heritability of 0.37 is really impressive. Some phenotypes related to vitiligo, melanoma, and speckling in the lower row. And this is the next highlight among the follow-up studies of the Copernicus project, a study of Barbara Wilder and co-workers on the Y chromosome in horses in general. Y chromosome in horses is kind of special because in all other species, mammalian species, you see a reasonable variation in Y chromosome, but not in horses. In horses it was extremely difficult to find genetic variation using more powerful, newer methods. Barbara succeeded in this endeavor, and she could show that there is certainly more than one haplotype on the Y chromosome. And she brought a neat example of how a new mutation from the British thoroughbred can be spread through the population when the traffic with breeding animals is vivid enough. But I'm sure Barbara will tell much more about this in her talk today, so I will not stay with these slides for longer. The next, maybe not really related with horse genetics work, is identification of a splice signal in horse genes for milk proteins. This was a student of mine, Tina Lenassi, who discovered that the signal in the equine beta-casein gene in the intron 1 is really a universal and strong signal which can trigger splicing also in human cells. And but certainly she worked on samples from lipizans, so this brought lipids in horses also into the context of gene splicing, which is another important topic in research. And we speculated at that time, it was an actual modern speculation to say that splicing of DNA is not post-transcriptional, but is co-transcriptional event, and this changes paradigms in thinking about how mRNA is processed in the cells in general. So this was more of the general contribution to genetic research. And then we did some more studies on variability of casein genes in horses, because we were at a certain moment interested in horse milk as a dietetic product, and we are still working in this field, certainly using lipids in mare milk. And finally, I would like to thank all the partners in the project, especially Thomas for organizing this very nice event, but the other partners mainly for stimulating and nice discussions we had during the project. Certainly also directors of the stats who enabled these visits and agreed to collection of the materials and use of these materials for research purposes, And finally, our supported from the financial side, mainly European Union, but also local research agencies in Austria, in Slovenia, in Switzerland, who supported with additional funds work on this project. And for this, I thank you for your attention.